There are a number of challenges that we face in diagnostics in the developing world. The infrastructure in terms of simple things, air conditioning, electricity, the distance that a patient may have to travel in order to get a diagnosis. We're looking to identify some very unique technologies that we'd be able to apply to the challenges that we face.
This program's organized by having groups of researchers that are in different topic areas such as sample preparation, amplification, and transduction. And as a result of that, we expect to see the development of best-in-class solutions in each of those areas.
Our project is focused around collecting a painless blood sample. We have a microneedle array, and that microneedle array is inserted into the skin at a very high velocity. So that gives us a painless insertion. And then the device itself is all fully contained, so the device can be placed on the skin, press a button, the sample gets collected into the device.
We want to make simple chip that allow us to just drop the blood and then separate this blood with the plasma as well as a red blood cell and white blood cell on one chip.
Our project uses a method of heating and cooling samples so that you can identify the pathogens of interest. What we're trying to do is make this technology available to the wider application of a point-of-care setting, which means that it has to be inexpensive, it has to be robust, and we also have to automate as much as possible.
If we take a blood sample from a patient, we need to process that sample to get the important parts out of it. And what we've done is learned how to create very stable interfaces between oil and water. And by moving parts of the blood sample through this interface, we can separate parts that we're interested in from parts that we're not interested in.
We're trying to develop a technology to remove the biomarkers from a patient's sample. We have a single length of tubing that's self-contained, and it uses processing via magnetic field to transfer particles that are bound to the biomarker from one processing solution to the next.
A patient infected with a bacterial or a viral disease will often be diagnosed by the presence of a protein biomarker in their blood or urine or saliva. That's typically done now using antibodies, but antibodies have a number of drawbacks. And so we're trying to develop our X-aptamer technology to replace antibodies.
Oftentimes tests that go out into the field use antibodies to detect those signs of disease, and that makes those tests unstable. Tests have to be stored under some refrigeration. And so we are trying to replace those antibodies with things that perform the same, but are very stable and can be stored in the trunk of a car on a hot day.
Oftentimes, the molecules we're going to be looking for in the sample are present in such tiny, tiny amounts. You need some way to detect them. That molecular amplification process, which my lab will work on, is meant to identify the very few molecules that are present at the outset and to make the signal from those molecules grow to the point that it's easily read at the end of the test.
Our project is to use DNA as a polymer to self-amplify signals for point-of-care diagnostics. In particular, we're trying to create a low-cost, battery-powered, enzyme-free. Instead, we're using light to amplify the signals.
The goal of our Grand Challenges Point-of-Care Diagnostics project is to make a palm-sized battery-powered readout device. What we are hoping to do in our project is to leverage microfabrication technologies and basically reduce the size and the cost of those readers.
My hope is that we will come up with a device, a diagnostic point-of-care test that is an open platform that will be utilized by a variety of people to test a wide assortment of conditions and ultimately have a major impact on healthcare in the developing world.
We've been very fortunate to be able to assemble a group of grantees that have some really revolutionary technologies that, if successful, we think gives us a unique opportunity to be able to take novel approaches to science and diagnostics